Good afternoon, everyone. So I'm going to give you an overview of the flow cytometry shared resource. So this is the team. Myself, I've been a director for a few years, I think since 2017. And Luke, shown here, is the technical director, is primarily based at the PNS building and is overseeing the day-to-day operations of the core. Uh, those who are in ICRC may know Steve, who is the associate manager and based at the Cancer Center ICRC building, overseeing the management there. And finally, 1E with an additional staff working at the core. So together, we have really, really many years of experience in various fields of science, immunology, cell biology, stem cells, oncology, and so on, and primarily also on using flow cytometry and cell sorters. So the services that we provide are, of course, the maintenance of instrument fluid daily calibration, maintenance. We provide troubleshooting of our users during regular hours. And we provide also operation of all cell sorters upon request, although some of the cell sorters are automatically with users, with uh, allocated staff because of the complexity of operating these sorters. Importantly, we provide also free consultation and training for all these instruments and also help with the preparation and experiment design. If you have any complex experiments we're using flow cytometry, we can help you with that. Also, but less usual, we also help with data analysis. So these are all the instruments that we make available to our community. They are, as I said, situated in two different sites. The PNS site, room 503 and 514, that has all these instruments available, and the ICRC site on the third floor. So those are highlighted here in purple are the newest additions to the core in the past year. So we received a 5-laser Cytek Aurora sorter that we're very excited about. We also have, at least for the time being, a Fortessa 5-laser Fortessa that was brought with Dr. Sutherland, currently in the core and shared with us. And then each 5-laser Cytek analyzers were also recently fitted with a sample loader to automatically analyze both tubes and plates. I'll tell you more about it in a minute. So these is, first I'm going to talk about analysis and analyzers. So we have a number of conventional analyzers, but for those who have been with us for some time, you know that over the last few years, we have really equipped our core more and more with spectral flow cytometers like the Aurora, and these are extremely popular and extremely powerful. I'll explain you in a minute why, but basically now I would say almost 50% of our usage is on spectral flow cytometry. So for those who are not too familiar with these, with flow cytometry, in conventional flow cytometry, for each fluorochrome that you use, you detect the signal that is emitted through a single detector. And that detector is a signal that is also stimulated by one laser. So you basically analyze something that comes from a single detector. In spectral flow, for each fluorochrome, you detect the signal emitted through 5 lasers, up to 5 lasers, depending on the machine, and through many detectors. For example, for a 5-laser Aurora, you have 64 detectors that encompass most of the spectrum, the light spectrum. And basically, the signal that you see, for example, with our one analyzer is only a partial feature of the whole spectrum that you can analyze by by spectral flow. So typically the highest signal what you see is what is being picked up by conventional flow, but in spectral flow, you analyze a lot more features, a lot more subtleties of that emission signal. And same thing here, for example, with PE, the signal that you see here is there, but there are a lot more signals that can be picked up. So if you're already using conventional flow cytometry and you really want to push your limit and thinking about using spectral flow cytometry, you can basically do the same thing using the same analysis program. Everything is the same. You can keep the same panel you used to use and just add additional markers. And it's very powerful because even markers that were initially considered interchangeable in conventional flow, like APC and Alexafluor 647. These would be, you can use one or the other at the same channel interchangeably, but you could not use them together. In spectral flow, and you see here, this is the signal that you would pick with spectral flow, now relatively similar, and it's impossible to differentiate them. But with spectral flow, you can see a lot more differences, and that's sufficient to allow you to use both fluorochrome together in the same panel and still differentiate the signal, even if the signal is on the same cell, which is extremely powerful. So here we have examples of cells that are double positive for APC and CDAFC 647, and this is just as good as having APC versus PE. And so, and another benefit of using spectral flow is you can apply, you can better separate the signal that are is non-specific versus specific in large autofluorescent cells. I'll give you some example. But in general, again, for those who are new to flow cytometry, I just want to stress out a few basic notions. First, that flow cytometry is a single cell analysis, and it's mostly proteomic. So you measure a lot of things related to protein. So things like surface markers, intracellular cytokines, transcription factors, even phosphoproteins. So you can look at also cell signaling with this technique at the single cell level. And not only looking at the expression of cells expressing that marker, but also the relative protein expression at the level of these expression. And it's also possible to even stain certain RNA. And in this case, you can even correlate protein versus RNA for a number of targets. And there are also many different types of functional measurements that can be done with flow cytometry, including cell proliferation, looking at number of divisions, cell cycle. You can look at signaling like calcium signaling. And also protein phosphorylation I mentioned earlier, apoptosis, endocytosis, oxidative stress, certain activity of certain enzyme like ALDH, just to name a few examples. Other things that can be done include protein-protein interactions, and also we can do high throughput screening, including CRISPR screen, and that also involve typically cell sorting thereafter. And I think some people not often think about time as a measured parameter in flow cytometry, but that could be very important. If you measure changes that occur over time, typically in conventional flow, you would see, you would have to pause the measurement in order to add a substrate, for example, and then restart, and you can see a sort of a break here, and it is not very continuous. So an advantage of the 5-aura laser, the aura in general, is that it is not requiring pressurization of the tube. So typically, once you insert the tube, it's sealed, and in conventional flow, it's pressurized, so you cannot add anything without actually stopping the system. But with the Aurora system, it's not pressurized. So even though it's sealed and you can't really access the inside of the tube to add a substrate, you can very easily make a hole and add a substrate at any time. And because it's not pressurized, you would not have to interrupt or pause the acquisition of your samples. So that's quite interesting. I mentioned about autofluorescence extraction. So that's very useful in case where you have large cells that have a high autofluorescence, and at the same time, you're measuring a fluorochrome that have a weak signal. So in this case here, we're looking at a staining actually of an mRNA that was amplified. The signal was amplified with Alexaflor 488. And the only difference in signal is found in this very channel here. Everything else is the same. Like all the signals you see here corresponds to the background signal of that cell. The only difference is here. And in a conventional cytometer, what you would see is something like this, very impossible to separate the signal that you see here. But with autofluorescence extraction, now you can see what cells are actually expressing that mRNA versus those that don't express it. Another examples here are gut epithelial cells that express the venous reporter. In conventional flow, you have cells that are positive, but everything is kind of mushed together. With using Aura together with autofluorescence extraction, you have a very nice separation of all those signals, allowing you to analyze this particular population. And until recently, it was not really very hard to sort those cells, but now with the 5-laser Aura sorter that we have acquired, it's also possible to get that great resolution and at the same time then sort cells that gives you that problem of low resolution. So with spectral flow, you can go up to a large number of markers. Here is an example of a panel that uses 40 antibodies, and that allows you to detect 45 metaclusters or discrete population within all immune cells, including myeloid and NK cell, B cells, and different types of T cells, very high precision. So it's similar to what you would do given the high parameter aspect of this analysis using a large number of antibodies. You can do something similar to what's done in single-cell RNA sequencing, where you using UMAPs, for example, you can identify discrete clusters of cells. And with one image, you can have a very nice overview of where your particular marker is expressed, for example, which would take a lot of different panels to do that with conventional flow. So let me give you some examples from actual users. In these examples, this is from the Moore lab in a recently published paper where they were looking at the effect of using checkpoint inhibitors in tumors. As you might know, the use of checkpoint inhibitors can lead to immune-related adverse events, such as autoimmune reactions. And in this case, as the treatment with the combination of anti-PD-1 and anti-CTLA-4 can lead to a significant reduction in the tumor size, but at the same time, you can have an increase of T-cell infiltration of a number of tissues, such as liver, lung, heart, and colon. And in this paper, they use a 33 marker panel with the Aura, 5-laser Aura, and studied the T cells in the tumor as well as in the liver in this example here. So this is to get the clusters you have with the combination of the T cells from the tumor and the liver. You have the CD4 cells here and then the CD8 cells identified in this side, on this side. So again, just like in single-cell RNA-seq, you can identify clusters with different cell population, and the more markers you add to your panel, the more precise you can be in identify these populations and phenotype them. So here we have a 12 clusters of different population of CD4 and CD8 T cells that either infiltrate the tumor or the liver. And then here you can see they are, many of them are found in both types of tissues, but you can identify particular clusters that are particularly and significantly enriched in the liver, as opposed to other clusters that are particularly enriched in the tumor. So that's, again, just looking at just a few figures and not even fax plot, you can have a very clear vision about what is going on in this study. This is another example from the MAPARA lab, again, using high-dimensional spectral flow analysis here with 24 color, and which allow them to analyze a large number of immune subsets that are listed here. And in here, there are immune cells that they use for bone marrow transplantation. And the idea here is they want to get rid specifically of CD45RA positive T cells, which are naive T cells, and primarily responsible for causing GVHD in the recipient And so the problem is if you do a regular depletion of cells using anti-CD45RA, you get rid of not only these naive T cells here, but also all the B cells, NK cells, and a population that appears to be plasmacytoid dendritic cells. And while these ones are problematic, the other cells are actually quite useful if you keep them to prevent GVHD. So in this assay here, they tested the different types of depletion, called oligonucleotide cascade-based depletion, and you can see very clearly that these naive T cells are specifically deleted while sparing the B cells, NK cells, PDCs, and of course the stem cells. And then finally, I just want to mention about another study. This one is in xenotransplantation, so this involved different species. So this is a porcine to baboon model. And so as you have two species, you need even more markers, because here you have to combine pig markers and monkey markers all together. And this is using a 22-color flow cytometry panel. And here they published that panel showing how they have optimized that panel to identify the different cell populations. Just want to mention that these OMIPs are something that are very useful if you work on a particular area of research and you're starting from scratch. I suggest that you go look for the OMIPs search for OMIP. And they have already published a large number of complex panels that are used for certain populations. These panels are already well validated. And so it's a good way to start with these panels and then make some changes according to your project. But it's really useful resources that I highly recommend. And you might be able to find a OMIP that fits you. So a few words about our cell sorters. The original cell sorters, so the BD influx, are still in functional. So these are the most complex ones that are strictly operated by our staff. The BD area is one that has reached its end of life. So it is still operated. It's still under service contract, but it won't be for too long. And right now, these are the two newest instruments. I mentioned the spectral sorter or a sorter. And I'll give you a little bit more information about them so that you can make up your mind about which one would be probably more suitable for your research. So basically, both the BD and the Sony instrument are relatively similar in the way that they have both for lasers, similar size nozzles, and they have basically the same capabilities, except that they are quite different in the complexity and the speed. So the BD influx is the most complex. You cannot use it by yourself, but it gives you very good compromise of speed and with viability. It's definitely the fastest sorter that we have. If you have a lot of cells and you have limited time, this is the one to go after. In contrast, if you have time, especially you have limited access during the weekday, or your availability of samples are such that you require time to process them and require access after normal hours, the Sony sorter may be a better one for you because you can access it yourself and operate it yourself. It's highly automated and easy to use, but it's the slowest out of all instruments. But because, again, of the after hours availability, you have a lot more time on your own to operate it. And then finally, the Cytec Aura CS, which is in a PNS building, is by far the more powerful. It has 5 lasers. And in terms of speed and automation, it's kind of between the other two. But again, if you're already using spectral analyzers and you already have your cell of interest and you have a panel of antibody that works well, it's very, very easy to just switch to go on this machine, use the same parameters, same panel, and everything will be exactly the same as you've seen in the analyzer, but now you are on a sorter and you can sort your population of interest. So we are coming to almost the end. I just want to say a few words in terms of software. So right now in the last few years, we had a new software that we've been licensing together with the stem cell cores called EasyPanel that allows you to design very good panel for complex analysis for any panel more than 15 or even 10, any number of markers you want. You can use this instrument, this software, to design an optimal panel that will take into account the type of markers that you use and the brightness of the fluorochrome that you use to maximize the signal that you're going to get at the end. And it also have a stock manager option. So that's very cool because if you use it, you receive antibodies, you can take a photo of your tube. It's going to recognize the catalog number, is going to put all the information on that database for your lab. And as you then build your panel, you can instruct the software to look into your inventory first and take advantage of the antibody that you already have. And then if some that you don't have, it can make suggestions about other antibodies that are missing or that you need to get. So this is a tool that is available for free for any Columbia users, if you have a Columbia email, and that our users find very useful. You can see that FCS Express have recently been acquired by Dotmatics, which is a large company that owns other tools that you might have been using, like Prism, LabArchive, SnapGene, and EasyPanel, and FCS Express. So FCS Express is the equivalent of FlowJo. They both are software to analyze flow cytometry data. And FlowJo, you can get the license from the microimmuno core. FCS Express, you can get the license through us. So we provide the site license for FCS Express. Just want to mention finally that on April 17, we have a informational learn and lunch seminar in person and also on Zoom, I think. So you can get information. You can actually, if you're not already a user of our core and therefore you would not have received the announcement, you can contact me. I'll show you my email on the last slide. It's going to be 1 hour and a half with lunch provided, and it's going to cover Easy Panel, but also Omic, which is another functionality provided through Dotmatics for related to cytometric analysis to help make your analysis more smooth, and particularly when we're dealing with complex sets of data. With that, I'd like to thank you all for listening. And again, this is my email. If you have additional questions or you want to receive the announcements for that particular seminar upcoming, just email me and I will forward everything to you. Thank you.